I have my interview today and it was supposed to be from 2 p.m. and I joined it at 1:30 because they told us to log in 30 minutes before the actual interview time, and they sent a message that you join at 3 p.m. So I have 1 hour extra to feel underconfident and overthink everything. Either way, this is the setup. I was just reading the things that I've done. So this is one kind of webinar I attended, which was very advanced bioinformatics, and I don't remember much from it, but yeah, I've mentioned it in my CV. So this is the research groups that is present in the institute. I did some research, what kind of work is going on, a lot of research groups, and these are some of the articles that I found interesting, so I read them. So like I have like a basic idea of what the research was about. And I also found this. So this is my notebook from class 11, okay? And I drew this and this. So I had these set of, I think, oh, I also made flashcards, by the way. See? Yeah. Anyway, so I made this notebook in class 11. But I had a series of such notebooks. So the first one had digestion and absorption, and on the back page it had breathing, because in that notebook I made notes of those two chapters. So human physiology had, I think, about 6 chapters. So I divided it into 3 or 4 notebooks, I think, like this. And each one of them, this is basically recycled brown paper. So I drew it and then I wrapped it with cellophane paper. So much effort, I cannot even recognize this person anymore. 3 minutes to go. The interview went really shitty. So there were 3 people in the panel. The first professor asked me some questions that I was really good at them, and I was explaining everything, and then he still didn't get the exact answer that he wanted, while I had it all along, because it was just too obvious to me. But I ended up saying it at last, so that was good. But after that, there were 2 professors, so one of them asked me questions from microbiology, and I don't know, I studied so many things, but the questions that they asked was, for example, he asked me, how do you differentiate between a living cell and a non-living cell when you are dealing with the growth culture? And I don't even remember the last time I studied this, because this is something that you study in the first semester. And so I remembered that you just stain the cells, and for some reason I remembered the dead cells will not retain the stain, and the living cells will retain the stain. And I said that, but apparently I said the opposite concept, and so that got, like, the first question that he asked me, I just couldn't answer it. The question that he asked me, what specific channels does a bacterial, like, what are the similarities between a bacterial cell and a nerve cell or a neuron? And then he asked me what are the particular bacterial channels, and I still don't know what is the answer that he expected. And I moved on to the next question, which is this question, and I apparently gave the exact opposite answer. And then the third teacher asked me what is my particular area of research, have I searched any of the research groups, and I told her that the ones doing research on regenerative on C. elegans, because that's what I remembered. Like, there are 16 research groups, and I didn't. Yeah, and then she asked me technical questions, like, not technical questions. She asked me questions like, so why study C. elegans? And the problem with me is that I should have stuck to one research group and उसका पूरा बाल का खाल निकाल के पढ़ना चाहिए था. And what I did was I just not getting into this institute.